Hello, and thank you for attending our virtual presentation on biopharmaceutical applications of FID for HPLC. My name is Tommy Saunders, and I work for Activated Research Company, and we're excited to share some results from our Solvair detector. So Activated Research Company, or ARC, is a company that was founded in 2014, and we really represent ourselves by disruptive science for a better world with our mission to make chemical analysis easier and more accessible using our expertise in catalysis and additive manufacturing. We specialize in post-chromatographic reaction devices to make the detection of molecules more accurate and simpler. We're based in Eden Prairie, Minnesota, which is right outside of Minneapolis, and I've been at ARC for a little over 4 years now. My role includes applications development, technical support and field service, and research development for new products. As I mentioned earlier, ARC specializes in post-column reactors and detection systems, and the common theme is catalysis and simplicity. We have an amazing team to develop this technology and apply it to different areas depending on the customer need and customer feedback. Products include the polyarc and jetonizer for GC FID analysis, a custom combustion reactor for isotope ratio mass spectrometry, a GC by GC flow modulation system at low cost, and Solvair for HPLC, which is what we'll be discussing today. And again, the common thread between all of these is based on customer feedback and pain points. We've developed these products to make things more simple and easier, and leaning into our niche expertise of catalysis to develop these products. So before we begin talking about Solvair and its applications, I want to give a little bit of background on the biopharmaceutical industry. Biopharmaceuticals are among the most revolutionary achievements of modern science, resulting in targeted products with fewer side effects. These biologic-based drug products can be comprised of sugars, peptides, proteins, and many combinations thereof. The market is growing rapidly, and advancement in chemical analysis is required with the development of biosimilars and new products to make sure that their processes and their manufacturing is adequate and safe for the safety of a customer and also for the efficacy of the drug product itself. Current biopharmaceutical analytical laboratories primarily employ HPLC, with the most common detectors being UV-based detectors such as the UV-Vis and the diode array detector, and the mass spec for quantification and process analysis. And those, although they're robust and well-developed detectors, they do have their drawbacks that come with them. Notably, the UV-based detectors do not detect molecules that don't contain a chromophore, so non-chomophoric molecules would be invisible to that detector. And quantification with mass spec can be pretty difficult due to a variety of effects that molecules may go through, including ionization, matrix effects, and compound-dependent responses. So this typically results in the more cumbersome task of doing targeted calibration, and that's sometimes unfeasible due to unavailable standards for calibration. And there are other detectors that are sometimes used, such as ELSD or CAD, and those do have problems though with their linearity and their ability to handle a gradient without any other additional compensation. So there's clearly, based on responses we've heard from potential customers and other people we know in the industry, there is clearly an opportunity for a universal detector, and there is a need for it in this space. So based on that feedback, we at ARC have taken our expertise and what we've done in the past in the GC world and taken a couple of years to develop the Solvair, which is a carbon selective detector for HPLC. And what it is we bring FID to the HPLC, and we're very proud of the product, and we're going to go through a few different results that we've gotten in the biopharmaceutical space, but also a little bit about how it works as well over the next couple of slides. So what Solvair is really is a universal FID for HPLC. So there are obviously some fundamental things you need to accomplish before running analytes through an FID with that are effluent from an HPLC. First and probably most notable is you need to completely remove the mobile phase. If you don't do so, you're going to have problems with your FID, either keeping it lit or saturating it with the constant flow of an organic solvent, for instance. So we, with Solvair, have been able to accomplish this, separating the mobile phase out and maintaining the non-volatile analytes in the system for further detection. And we also, by doing so, have an analyte response that's independent of the mobile phase composition. So whether you're running a gradient or isocratic with different solvents, you don't need to account for that in your response factors, for instance. So once we get the non-volatile analytes removed from the mobile phase, we then convert them into methane for a universal FID response, similar to what we've already done with GC. So this is where our catalysis comes into play, and we use proprietary technology to get these non-volatile analytes into the gas phase and converted to methane. So once we've done that, then we have at least a 5 order of magnitude linear range, going down to a limited detection of roughly 10 ppm for standard injection volumes. And the device is standalone and automated. So it's simply push play to start. All you need to plug in the front is a transfer line from your HPLC, and we run an analog to digital converter in the back for your signal, which is all processed with your CDS. So we like to share this schematic to illustrate the detection concept for Solvair. And you can see on the right as well, for illustrative purposes, the size of Solvair relative to a 1290 stack there. So what happens is that HPLC effluent goes into the detector, where we first remove the solvent or the mobile phase from the sample and retain the non-volatile analytes, and those subsequently get converted into methane for that universal response in the FID. Substitution on molecules like oxygenates or other heteroatoms, such as halogens, for example, will reduce the response in an FID. So it's important that when we get the product to the FID, ultimately it is methane for that universal response. So as an example, we can look at sucrose present in a sample with water and acetonitrile. So the first step in Solvair, once it exits the HPLC as effluent, is to remove the mobile phase, so the water and acetonitrile come off, and you retain the sucrose in the detector. Next, that detector is used to convert the sucrose into 12 methane molecules, which eventually get detected in the FID in a universal fashion for every individual molecule. Sucrose is a very important molecule in the biopharmaceutical space, with applications in stabilization, supplementation, and preservation, for example. With Solvair, we can detect this linearly with a portion of the linear range shown here. Detection of non-volatile species with an FID like this is very exciting and opens up many possibilities in the biopharmaceutical space. Maltotriose and maltoheptaose are two other sugars which have large impacts in the biopharma space, particularly in conjunction with enzymes such as alpha amylase. Universal carbon detection is shown here on the area versus carbon concentration plot on the right. Molecules here on the plot range from 12 to 42 carbons per molecule and contain many oxygenates, which typically would reduce the response in an FID because of that substitution. But our catalytic conversion to methane assuages that to give you a universal response in the detector, and that's seen here on the plot on the right. So bovine serum albumin, or BSA, is an example of a protein widely used in the biopharmaceutical space. Here we show it can be detected in a similar fashion to the sugars using Solvair. Detecting a molecule as large as this with an FID is unheard of, and this will offer much simpler protein quantification methods in the biopharmaceutical space. So some of the applications that bovine serum albumin is used for include ELISAs, immunoblots, immunochemistry, nanostructure synthesis, fetal bovine serum, and the list goes on. And it's really excited to see a molecule with this much molecular weight, 66,000 Daltons, detected in a very similar fashion to other molecules with an FID. So here we show a universal response for the sugars and the protein using Solvair. Because of the conversion to methane, one can now accurately quantify a sugar such as maltoheptaose against a protein, reducing the need for calibration runs and the need for obtaining expensive standards, sometimes prohibitively expensive or unavailable overall. The equation shown here illustrates how the conversion to micromoles of carbon is done. You simply just need to account for the molecular weight and the number of the carbons present. But it's really exciting to be able to look at a molecule such as sucrose with 12 carbons per molecule and compare it directly to bovine serum albumin with over 3,000 carbons per molecule and still depend on that accurate quantitation. So after looking at sugars and proteins, we wanted to take a look into peptides as well, because they represent some of the most prevalent compound classes in the biopharmaceutical space. And we wanted to show accurate detection and quantification on Solvair without doing any calibration. So we selected this quantitative standard, which is a peptide mapping standard from Agilent. And it contains a variety of different peptides with different peptide sequences and different numbers of carbons per molecule, as you can see in the table here. And here we see the chromatogram output of Solvair for the peptide mapping standard. All 10 peaks are detected, and you can see the method below indicates there is a gradient over 25 minutes going from water containing formic acid to acetonitrile containing formic acid. And we've been able to maintain the separation done by the HPLC with our peak shapes and narrow peak widths, and our minimum detection limit here is about 11 ppm. These peptides were quantified using one internal standard without any additional runs, and you can see fairly accurate quantitation. The small variations shown in the results are most likely attributed to losses in our chromatography system, which isn't bioinert. The plot on the right shows the carbon-based response factor for each compound in the sample, and to give you some idea of where the errors lie for each individual compound. So we're really proud of this result, that this detector is able to give you such a good quantitative result for a standard, knowing that there may be losses in the system or in the sample itself, and you don't need to do any calibration to understand that. So in an effort to compare these peptide results to another compound we have, we wanted to look at the much larger protein in bovine serum albumin, which has the molecular weight of 66 Dalton and over 580 amino acids. And the results, as you can see in the chart on the right, are comparable when you look at a molecule as small as the first one there with a 7 amino acid sequence, and you compare it to such a large molecule as bovine serum albumin. And then the plot on the left also shows you this in terms of linearity, displaying the linear range of Solvair, but also how you could extrapolate the curve and still accurately quantify protein relative to the information you might have about a peptide. So we've been talking mostly about, exclusively about non-volatile components so far, but I do want to illustrate a little bit as to what happens when you put a volatile compound or semi-volatile compound in Solvair. So we do only market the detector for universal response for non-volatiles, because once you start to have a vapor pressure for the molecule, you do start to tail off on your responses, and that's because of our thermal evaporative technique in addition to other variables at play in the detector. The molecules shown here all have a greater than 400 degrees Celsius boiling point. So you can see there's some detection there happening, but as you start to decrease the boiling point, you then really get losses that result in either very, very high limits of detection or very difficult and irreproducible analyses. So, in conclusion, we have shown that Solvair can be a very powerful detector for analysis of common biopharmaceutical compounds, including sugars, proteins, and peptides, and you can do so with a universal carbon response, allowing for a single point calibration and universal detection. This is only possible because of the detector's ability to separate the mobile phase from non-volatile analytes, convert analytes to methane, and detect them over a wide linear range. So thank you very much for your time. I'm happy to take any questions on the Solvair or our general product inquiries you may have, and I hope you visit ARC's virtual booth to learn more. There is my contact information below on our website if you want to visit for more information. Thanks again.